Thank you for giving me this opportunity. I'm Graziella from the Aerospace Technology and Science Department from Brazil, and I have a question for Dr. Laura Marcu. How do you control the depth of the fluorescence spectroscopy technique, and what does the neurosurgeon do, for example, during surgery? Does he remove the top layer and start digging underneath? And how does this noise of blood cells and environment lighting affect the spectroscopy? Thank you.
Okay, so first the penetration depth was the question. So the penetration depth for this light, it's about 250, 300 micrometers, depending on the tissue optical properties. So, however, during surgery, they are interested on removing residual tumors. So the chunk of the tumor typically is removed. So the idea is to have relatively limited penetration depth and to sense changes shallow changes at the surface to continue to remove and then stop when you have normal tissue, especially if you want to increase the accuracy of surgical resection. So in terms of blood and the presence changing at different distance, fluorescence lifetime is minimally affected by the changes in intensity and the presence of different absorbers. Provided that you have enough photons, then you can quantify the fluorescence decay. You cannot rely on intensity, of course, but the reason we use fluorescence lifetime is because of this disadvantage. And of course, you can remove the blood if it is in excess by either flushing or a suction, so there are ways of doing it. Was any other, did I miss any?
Hi, my question's for Dr. Maria. In the BRCA negative, BRCA positive population, you have, you did the exome sequencing, and you can account for quite a bit more in terms of the mutation, the load as possible driver mutations. Are you considering doing de novo assembly, whole genome resequencing on the positive population? Because the positive population, these are affecting DNA repair, double-stranded repair. So you're more likely to have rearrangements. So are you considering taking that population and looking for microinversions and other types of rearrangements that may also serve as additional driver mutations that could be driving the progression of these BRCA1 positive tumors?
Okay. Do you mean in the normal and the tumor tissue of the women that has triple negative with the BRCA1 mutation? Yeah?
Yeah.
We don't do, but we think in doing because what we are detecting now is, for example, in the normal tissue or even in the tumor tissue, we have more mutation with a low frequency in the women that has BRCA1 mutated. And maybe this difference, we can also have some rearrangement. But we
inside
have to
the
do
cytogenetics,
whole genome. And,
yeah, with whole genome you'll find the smaller rearrangement, but
yes.
Cytogenetically, do you find higher numbers of rearrangements in the triple negative BRCA positive population? Just by cytogenetics, do you see greater?
We have some CGH array data, but we are not, how can I say, we saw some difference. In the triple negative with the BRCA1 mutate, we saw like a serrate. It is just not a high amplitude gain or gain. actually. But just we have some data.
Yeah, it would seem
And
that that
it's
would be very
different.
destabilizing having because you found reduced expression of the BRCA gene in those cases, right? That function.
In the wild type, yeah? In the BRCA wild type,
yes. So
but we have to confirm. We just have a hypermethylated BRCA promoter in the, and we are doing a confirmation in the DNA and the, just to be sure, because this is a whole analysis on whole DNA analysis, and we are doing the confirmation, and we are also doing the gene expression evaluation, just to be sure that the BRCA1 in this case are downregulated.
Yeah. But
Yeah.
then you would expect higher numbers of
Higher number of samples.
and you expect a higher number of rearrangements because you're not
Yes.
repairing. So I think doing the
Yeah,
whole
that's a good
genome
idea.
resequencing could be very informative
Yes,
in these
yes.
cases
Yeah,
for
sure.
for rearrangements, structural variants.
Maybe FAPESP can
Yeah,
fund
that's a good one.
us.